Welcome to FYI, the For-Your Innovation podcast. This show offers an intellectual discussion on technologically enabled disruption, because investing in innovation starts with understanding it. To learn more, visit arc-invest.com.
ARC Invest is a registered investment advisor focused on investing in disruptive innovation. This podcast is for informational purposes only and should not be relied upon as a basis for investment decisions. It does not constitute, either explicitly or implicitly, any provision of services or products by ARC. All statements made regarding companies or securities are strictly beliefs and points of view held by ARC or podcast guests and are not endorsements or recommendations by ARC to buy, sell, or hold any security. Clients of ARC Investment Management may maintain positions in the securities discussed in this podcast.
So, this was really fascinating conversation that we had with Umoja team. Really for me, what was really kind of true paradigm shift is, not only their technological innovation on actually building in vivo cell therapies, or how they call it, a living drug, but also the scale that enables you, and also in addition, there are few layers of the scale that you can have there, but also a huge diversity of the different diseases and conditions that you can be going. So there's a compounding effect here that I can see it, and I think the other point here is that how does this really change the traditional pharma drug discovery model, which is very done linearly with a huge amounts in the R&D, and then manufacturing is the end. Here, if we build the initial product that that's a platform, we can have multiple thousands of the different products coming from there, almost like a SaaS business, but in the just a world of atoms, right? And that then the manufacturing, that part of the equation becomes much more important than the R&D cost in the drug discovery. How did you where did you see the biggest inflection point in this conversation?
Yeah, look, I think it was very interesting, and the leadership told the story really nicely. I think, to me, as you say, the scale up is very interesting. I was, really interested to revisit the investment case, essentially, and why this is such a compelling go-to-market to ultimately help patients in a major way. And, one of the huge things is just the cost declines that one can see with this kind of technology versus the current generation of CAR-T and related approaches. I think, even though the in vivo approach hopefully is more programmable and has more flexibility maybe than some of the ex vivo approaches, although, some of them are great too, what I hadn't fully appreciated is that flexibility, which often comes with increased expense, actually looks like it will come with a massive decrease in expense in this case. So really opening up the application areas of this, the scalability of this, and hopefully providing access to many, many more patients domestically and globally, which is very, very exciting. And I think the ability also to go after solid tumors beyond the blood-borne is a very, very interesting area. And I know others are working on it with different approaches too, but the fact that that's well within the remit here, because there's so many of these unserved diseases that could be amenable, and the immune system, blood gets everywhere, the immune system gets everywhere. And then beyond that, thinking through the outside of oncology, the application areas. So within other disease areas, I thought it was very interesting around talking about autoimmunity, particularly in the way that could be modulated, and then even ultimately into longevity itself and the way that immunosenescence, potentially, this technology might have some application areas. Because I think it's one big piece we sometimes don't know when thinking about this, don't factor in. It's just, for instance, with the immune system, quite how, if you take a first principles approach to longevity and thinking about the way that health goes wrong, you could almost think, well, there are lots of things in the body that are fairly static. They're not even cellular, right? Like laid down bone, for instance. There's obviously cells in bone, but some of the bone, part of the bone is not even cellular anymore. Then you've got this enormous spectrum within the cells and so the acellular stuff degrades at some rate and obviously can get replaced by the cellular component that is there to replace it. Then you've got this incredible spectrum of different length of life of different cells. So from neurons, which live essentially a whole life, or even some of the cardiac cells live up to or often a whole life or at least most of it. And then the other extreme is some of the immune cells, so some of the white blood cells live only 2, 3, 4 days. So you've got this sort of 10,000-fold multiple between living for 30, 40,000 days, right, like a whole human life, or 30,000 days anyway, to living, say, 3 days. And I think often we don't factor that in. And clearly because the immune cell part or other cells within the blood are driving so much of that cell turnover, that's a big reason as to why the immune system is so important in aging and disease, and of course in cancer, but also beyond that. So I think that was a big takeaway for me. Is clearly this is very applicable technology in oncology, which is the first port of call, but also thinking more broadly beyond that, and just really this is a company and a team that's really thinking about a broad and deep platform that can be used to address many different diseases across the board, but has a very clear focus to do something that's very a clear and present problem right now in the form of oncology.
Yeah, and for me also, there's a really big picture piece here. When you think about whole today's healthcare industry, which we call it a sick care at the moment. So originally, right, there is a disease, right, which is actually introducing some malfunctioning of the human body. And that malfunctioning leads to consequences, which one of the worst one is related to death. So our first approach was to try to reduce those consequences and just extend the life. Extend the life in terms that it's not shortened because of the disease. So we are just trying to mitigate some of the bad effects, right? And I think the most of the drugs is today in that stage. Then there is a next layer. Instead of just ameliorating some side effects and having that life not to be shortened, we can actually cure the disease. And so it's there is actually a bit longer life because there is no actual side effects. And then we go to in my view, the third phase, which is now it's not only about the curing, but it's actually enhancing capabilities to prolong the life. And you can see then here how the business models could be changing, as well. In this chronic management system, it's not about really, the incentives are not about the curing, it's about just maintaining that state with just some of the consequences, right? And so the incentive here is to have a patient's high volume, but still sick. With the cure ones, you cure the disease. The problem then becomes, well, how are you going to be making the money? So we're going to price the cures a lot that we can actually get the return on investment. When we move to this third layer, where it's a life enhancing, I can almost see it as a subscription service, because you just need to pay a little, but there's a huge amount of the people that could be using them and prolonging their life and being there for a much longer time. Do you see, what do you think about actually this parallel?
Yeah, I think, this is part of a bigger discussion that it would be great to have, and partly enabled by technology, enabling new models. But I think what you're getting at is also, there's a real need to step back and say, how would, knowing what we know now and having the technology we have now and the technology roadmap and what may realistically happen within the coming decades, how should health be paid for in a way that would move away from that sick care, as you say? I think there's an increasing and an interesting role for participants within the ecosystem that have maybe been involved, but maybe that could increase. So one could be life insurers, right? So life insurers are very motivated, stepping back and saying, on first principles, what are the incentives of each of the participants and players here? And when are those incentives broken or misaligned with others and with society and as a whole, and especially with patients? So life insurers, that's interesting because there's a lot of motivation to have people healthy and happy and productive and working and paying premium. So, just purely financially, there's a big incentive, and hopefully they care about other things too. I think there's also a big role for more the IDNs, the integrated delivery networks. So Kaiser's a very well known example, but there's also Intermountain, there's a whole bunch of others who have more of this sort of complete life ownership of the care cycle. And I think, there's some interesting thing to do, understanding there, because it's very different to some of the other systems where people are just moving around and switching insurers and switching employers and things, and being sort of lost to follow up and lost to care. And again, there's the chain of incentive, if you like, gets broken, as well as all the perverse disincentives that exist in certain parts of the healthcare system. So look, I think new models that drive incentives that move us away from sick care towards healthcare, clarity and transparency of incentives, including the ones that are maybe misaligned with other participants, especially the patient, and just better data, better follow-up, better tracking of patients, better integration of EMR. We spoke recently on a podcast with Eric Lefkowski, the CEO of Tempus, and we were talking about some of the things that they're doing around using AI to make with Olivia, their nascent product there, to give patients more control over their own health trajectory and health data, and to make some of that information more portable and therefore hopefully more usable through time. So, clearly there's a big role to play for those kind of companies like Tempus, who are integrating health data with at the whole patient level on the HR side, with also the biological data, and then working with those companies like Umoja, that we just spoke to, who are actually developing new therapies, and other companies working on new diagnostics. And just, I think we need to see better longevity or time series monitoring, better data, better understanding and use of EHR, and then more integration among and between, combined with novel systems or just reworked and adapted systems for paying for healthcare that identifies and tries to iron out those incentives or misaligned incentives to get towards that healthcare. Clearly a lot to do.
Exactly. And stay tuned. We'll be doing some research on this very, very soon.
Thank you.
Hello, and welcome back to ARC FYI, or For Your Innovation podcast. And today I'm joined with the Umoja team, Andy Scharenberg, who is the CEO, and Britton Russell, who is the CFO of Umoja. So recently, ARC has done the investment in the Umoja, private biotech company focusing on developing in vivo cell therapies. So maybe before we start talking about Umoja, Andy and Britt, you can tell us a little more about your journey and how did you end up at Umoja, and what are you actually doing there?
Sure. Nemo, maybe I can start and I'll hand things off to Britton. So I'm a physician by training, and I come over from the academic side of the scientific community. I worked for a long period of time at University of Washington and Seattle Children's Hospital, where I served as a pediatric immunologist, and spent most of my career working on technology for repairing gene defects in children who had inherited diseases of resistance to infection. So these are things like the bubble boy is a classic one in I would say the kind of public genre of knowledge. And over the course of my career of working on that technology, we developed a lot of really interesting ways of manipulating genes and delivering genes. Became very familiar with that. And one of the things I began to realize, so I'm 61 years old now, and developed a lot of interesting technology. And over the course of my career, I realized you can discover technology in the academic realm, but it's very hard to develop drugs out of it. And as I approached, the end of my career about 5 years ago, I also realized that, there hadn't been transformative practice-changing ways of treating patients that had really reached into my medical practice. And I wanted to be a part of developing something like that. So Umoja was in part talking to a lot of some of my friends and colleagues about, hey, what can we do at this stage of our careers that is absolutely transformative, and looking for a problem that I felt there was technology available to address. And so the name Umoja, the company, actually is a Swahili term for bringing together. And it was essentially, let's bring together the right technologies, the right people, the right team to do something as transformative as we possibly can that's really practice changing. And that's effectively where our in vivo gene delivery technology, VivoVec, came from, and some of the other technologies that we're working on to try and treat cancer.
Yeah, thanks, Andy. I joined Umoja about a year ago and was just incredibly excited about the science and technology and the transformative science that the company's offering. And previously was at a company called Parse Biosciences, a single-cell sequencing company. And before that, Archer DX, and then later Invitae, diagnostics companies where we focused on MRD and genetic testing. So, and then prior to that, AstraZeneca and Genentech and corporate finance roles. Really great to meet you all.
Maybe where we can start now is just trying to kind of explain to our audience what are in vivo cell therapies and why this is like a third generation of the cell therapy, and why this is really could be paradigm shift when we think about kind of cell therapy overall.
One of the most important ways or changes in how we think about treating cancer that's emerged over the last 20 years has been, instead of trying to poison cancer with what's drugs called chemotherapy. And the way chemotherapy works is you're poisoning a cancer a little bit quicker than you are poisoning the rest of your body. And that accounts for many of the side effects that we associate with chemotherapy. Things like hair loss and the fog and essentially terrible feelings that people have when they take chemotherapy. So that's the old paradigm. And the new paradigm has been to understand that actually a lot of the benefit that chemotherapy provides is because your immune system is in your body and actually already fighting the cancer. And chemotherapy is slowing the cancer down just enough for your immune system to come up, to catch up. And so people began working on new ways of essentially making your immune system more active to reawaken it to fight cancer. And one of the ways that has worked best is an approach called CAR T-cell therapy. So a CAR for a cancer immunologist is a chimeric antigen receptor. And it's essentially a synthetic protein that we can put on a tumor-fighting cell called a T cell and direct that T cell to directly kill tumor cells. So when you have cancer, when a patient has cancer, what has happened is normally your immune system is able to identify and kill off cancer cells. But over the course of your life, tumors can find ways to neutralize the immune system, to hide from it. And that's when cancer starts to grow out of control and starts to kill you. So CAR T cells are a way for scientists in the lab to teach your immune system to recognize anew tumor cells and eradicate them. And these have been worked on since the early 2000s. And there were some advances made that allowed them to really begin to work well around 2010 or so. And the way these therapies are generated initially was to take T cells, these tumor fighting cells, out of the body, actually modify them with viruses to express this chimeric antigen receptor, or CAR, and then to put them back in the patient. And one of the more important advances to get this approach to work was you actually have to give a patient small amounts of chemotherapy to make room for these T cells to come in and do their job. And this process was initially pioneered in a variety of academic labs, and then eventually commercialized by 3 companies. So there's 3 or 4 of these types of therapies that are available now, but they all work in basically the same way. You have to isolate T cells, you have to ship them, you have to make a drug product, you have to actually characterize that product, you have to ship it back to the place where the patient is, you have to give that patient a bit of chemotherapy and infuse this drug product. So it's made patient by patient. And there's complex logistics involved in isolating the T cells, manufacturing them, and getting them back. So the drawback is, even though these work very well, it's actually very hard to deliver them to large numbers of patients because that process is very expensive. These are somewhere around $400,000 to $500,000 per patient to treat. It's about 100 days. If you get diagnosed with a type of cancer that can be treated with this technology, it's about 100 days between your diagnosis and when you'll actually get your drug product infused. And for the most part, we're only administering these treatments at major medical centers that have pretty sophisticated staff and the ability to manage some of the side effects. So right now, if you look across the United States, only about 1 in 5 patients that could get a therapy like this is actually able to access it. So that's sort of the history of CAR T-cell therapy. And the commercial products right now require that long period of time. So the fact that these work so well, that's actually one of the things that when we were founding Umoja, we were looking at. We have this transformative drug product, and only 1 in 5 people can get it. There's no other drug product that has transformative effects where only 20% of the people can actually get it. So that was one of the problems we were trying to address. And in between approximately 2010 and the present, a lot of groups had said, well, maybe what we can do is take healthy T cells from healthy donors, make CAR T cells, and have those ready right away to give to patients. So that's a form of cell therapy called allogeneic cell therapy. And probably 15 or 20 different companies have tried all kinds of different approaches to try to make that work. And I think what's been disappointing, if you're a patient, is we haven't found a way to make that work as well as this bespoke way of making it from each patient from their own T cells. The effects of the treatment when you make it from a healthy donor are just much more limited. The durability of responses and the ability to get really deep anti-tumor responses, we just haven't been able to achieve it. And that's probably because when you have somebody else's T cell, when you give it to a patient, that patient's immune system sees that as foreign, almost like a tumor, and fights it, tries to fight it off. So in essence, somebody else's T cells, they are swimming upstream to try to kill the tumor because the immune system of the patient, which is trying to fight the tumor, just not very effectively, is also going to try to fight these incoming new cells. We haven't found a way to make the patient's immune system and these incoming donor-derived T cells work together effectively. So what we've developed at Umoja, we took a step back and we said, well, if that's the case, is there a way we can develop drug products that we can deliver directly to the patient and create the patient's CAR T cells directly in the patient and have them function? And so that's really the fundamental basis of what Umoja is doing. We're working on a platform we call VivoVec that allows that gene delivery to occur. And those are the way to think about it is the patient by patient way of manufacturing is the first generation of CAR T cells. Allogeneic CAR T cells are the second generation. And in vivo is really, in a way, the definitive way to do it. You've turned CAR T cell technology, this transformative mechanism of fighting tumors, into something that can be delivered right away as soon as a patient is diagnosed. And everybody would have access to it.
Right, it's more modeling what happens in biology naturally, presumably as the body is trying to fight cancers, but needs a helping, more than a helping hand. But
exactly that. So the way I think about it, it's a little bit like a genetic vaccine. So when you have an immune response against something, your immune system has a set of cells that initiate an immune response. And what they do is they find a way to collect all of the T cells, these tumor fighting cells in your body, that could see and kill tumor cells and activate them and expand them so that you have an effective anti-tumor response. And what we're doing is we're taking years of studying tumor cells where we know, okay, we want to make this we want to have T cells target this protein, which is expressed on this kind of tumor cell. We're genetically delivering that specificity. So instead of collecting natural specificities, we're artificially providing a specificity for these tumor fighting cells to target a tumor.
with the in vivo approach, does that give you more or less or sort of similar levels of numbers of targets? Or does it open up target space or constrain target space, but with other benefits, if you like?
Yeah, by strictly defining it, one of the advantages of genetically endowing these cells with a specific anti-tumor specificity is you're able to control the level of specificity. So one of the things that people think happens with natural immune responses, they can be very good, where you have the ability to relatively specifically target a tumor. But you'll also find instances where there are patients who have tumors where an immune response has started, but it also creates immune responses that target properly working parts of your body. So the immune response becomes too broad and actually gets out of control and can harm a patient by targeting organ systems that are recognized when they shouldn't be. So we have some advantage in that by providing a synthetic specificity where we've really carefully studied it and we know how tumor specific it is to make a narrower response that's more specifically directed at the tumor. So that's one of the advantages of engineering that specificity. On the other hand, there's only so many really good targets you can identify. So there is a little bit of a limitation as well. So that's, I would say, you have tons of scientists working on finding new targets to target various types of tumors. And one of the things you can think about as we begin to build out our technology is we're starting with some of these targets that are recognized now. People are identifying new targets all the time. So we will gradually build up a broader and broader armamentarium. So that limitation that we're faced with now in terms of providing a narrow specificity is one that will eventually begin to disappear as we have more time to build up new ways of targeting tumors.
And maybe a kind of follow-up question for me here is, when we think, and how you're saying, kind of when you were giving us the kind of history, kind of chemotherapy or kind of small molecules, we are trying to basically with them demodulate the system. They're kind of very, very crude ways. When we kind of move to a kind of cell therapy space, it's actually kind of taking the cells which to actually modulate the system and the particular disease. And so what I see unique about Umoja is the way to actually do that modulation in actual context, in actual organism, and that kind of gives you it helps you much more with kind of scale and kind of manufacturing, versus the kind of previous kind of models. But then I'm also curious, because you are in vivo and you're modulating, particular disease, does this also gives you kind of additional ways that you can do some additional kind of controls? Can we envision about kind of like third, fourth, fifth generation of Umoja system where you have using multiple small molecules to kind of increase or decrease the dose response, or kind of how they are kind of which kind of antigens they're going. So I'm trying to understand that your kind of approach gives you more of engineering diversity, which you can later kind of implement. If you get me what I'm trying to say here.
Yeah, I think so. And you can see. So one of the ways I think about it is, if you look back at the pharmaceutical industry and the ways that drugs in the 1970s were generally pretty small molecules. If you look at the size of a molecule, we have a way to measure size of a molecule called essentially a Dalton. Molecules for drugs that were developed in the 1970s, for example, were probably closer to 500 Daltons. And then people started finding ways of developing larger and larger molecules. One class of those molecules are antibodies. So that's and other types of biologics. Those are larger molecules. Those are somewhere in the range of 100,000 Daltons. So much, much larger molecules. And where we are now with a cell, you would be, a cell is composed of probably millions of molecules of 100,000 Dalton size. So the sophistication of what a cell can do is much, much higher than what an antibody can do, and way, way higher than what a small molecule can do. So it gives us a very large engineering space to work with in a cell, all of those different functions. The other thing is cells have their own library of functions that are already programmed in. That's the way immune cells are already set up to fight tumors, to recognize foreign pathogens and things like that. So we don't have to rebuild all that from scratch. We can actually, by understanding how cancer-fighting immune cells work, we can know, okay, hey, look, they have the ability to expand when they need to. We don't have to teach them how to divide. All we have to do is reteach them how to recognize a tumor cell. And once they do that, all that intrinsic programming that has been evolved over many years is then engaged in expanding, creating an army of cells that now have the right specificity to kill tumor cells. It's really the idea of a living drug that's what's unique about CAR T cells. Now, the process of reprogramming that CAR T cell, that is really kind of the trick, the one of the core technologies. And I think what the audience might appreciate is we actually have borrowed mechanisms from viruses in order to enable that reprogramming. It's actually directly analogous to if you think about computers and computer viruses. So a cell is a lot like a small computer. It has a ton of intrinsic programming. And all the time over the course of evolution, viruses have found ways to put a little new piece of programming into a cell in order to replicate that the virus itself. That's just the same way computer viruses work. Computer viruses gain access to your system. They replicate themselves. They may send information back out of a computer to somebody, some bad actor in a different place. So biological viruses work the same way. They inject new programming into a cell, and for a virus, all of that programming is directed at making new virus particles. So for our technology, we're actually using biology that's borrowed from the study of the human immunodeficiency virus, or HIV. And what we do is we take out the parts of HIV which are directed to simply making new HIV particles, and we replace that with our own program that teaches that tumor-fighting T cell how to recognize a cancer cell again, how to become essentially very active, make more copies of the cell instead of more copies of the virus, to create an army of T cells that fight cancer. So we're building, very often you'll hear the phrase from scientists, we're standing on the shoulders of giants. We are standing on the shoulders of many giants, people who were studying all of the different biology of different types of viruses, the specific biology of HIV, how tumor cells are recognized, and all of, probably many armies of scientists over the last 20 to 40 years who determined the basic mechanisms through which immune cells function. And we're in the fortunate position of being able to say, hey, we understand how those pieces fit together, and we can take that knowledge and create a drug that leverages all of that and that we can deliver directly to a patient. So instead of having to do all this complicated manufacturing, we now essentially have a drug that when we deliver it directly to a patient, goes in, delivers and reprograms T cells in vivo, and they now have the ability to fight a tumor that otherwise it would be invisible to them.
Yeah, and for me, when I think about really, living drug, right, and kind of using the cell for that, is the kind of, given that, they're already built-in functions, you really can think about kind of modular engineering. What I'm trying to say, you can be adding many different kind of bits and pieces to kind of enhance that drug to be even better, right? Where the traditional small molecule biologics, they kind of have kind of one function and that's about it, and you can't really change much what you give it. Here you have a time control that you could be kind of implementing, right? And so I'm just curious, thinking, just ahead, what this kind of modular engineering could allow us. Obviously with Umoja, right? You have tumor tagging, so kind of tagging the tumor with a marker that then this kind of the living drug could kind of recognize and kind of eradicate. And then obviously we know the tumors are evolving. If the disease is progressing, you can be adding the new tags and kind of keep kind of helping the patients. But what could be some other kind of potential kind of modular kind of things that you could use? Obviously something about kind of dose effect, decreasing the doses. So how this how we go from this kind of living drug to actually a living treatment kind of concept.
Yeah, and I'll come back to this idea of how small molecules work, how biologics work, and how cells work. With a small molecule, the way to think about that is it also modifies the functions of cells that it touches. That's how all drugs work. But it's always working within existing programming of that cell. So a small molecule will touch one molecule in a cell, change its function. That's how a drug will work. With larger molecules, you have the ability to touch a bigger surface, so you can modify cells in ways that we didn't have access to, because a larger molecule can modulate, I would say, maybe larger protein complexes, or can modulate proteins where a single, the touch from a very small molecule is not able to create a significant effect. What we're doing with a cell is we're actually adding new programming. It's not that we're working just with the programming that exists and changing that programming. We're putting new programming in. So to your point, the first generations of what we're doing, we're putting in a chimeric antigen receptor, which reprograms a tumor-fighting T cell to directly see a tumor. We can, along with it, we can put in additional controls that provide drug-activated pro-survival signals that make our T cells last longer, so that they have more time to fight a tumor in addition to making them more potent. So we can add additional programming, and I certainly aspire to a world where you begin to think of a cancer therapy as a little bit like an iPhone, right? Where your first generation has one set of capabilities, you can add to that. Each generation will have more capabilities, And that will provide a physician with more, more power and more control over that power in terms of how they're able to think about treating a patient with cancer. So that's for sure part of the future of Umoja. Right now, we're in the very first part of it, and I think one of the things I always like to emphasize is like the limit. We would love to create the most sophisticated treatments in the world. Generally, though, we want to make sure all patients have access to those, right? So the first generation of what we're doing, we focused on two things. One is the technology that allows us to do this reprogramming. And the second part is also building out the ability to manufacture this kind of sophisticated drug product at scale. So we spent, in addition to building the technology for that allows us to deliver, and again, in future generations, certainly we'll be able to add levels of sophistication. We've also spent a lot of time and effort. In fact, we've built our own manufacturing facility that's in the Boulder, Denver area that allows us to manufacture this kind of product at a cost where we think instead of treating 1 in 5 patients, we actually aspired to allow every patient that could be eligible to receive this kind of drug product. And one of Britton can maybe touch on some of this. So that manufacturing facility in Boulder, when you're manufacturing sophisticated drugs like this, one of the things, and you want to try to do that economically, one of the things you have to pay attention to is all the little aspects. You look at a complicated process, you have to address the cost of every little part of it. And one of the reasons that I think we were excited to bring Britton to Umoja is the background he's had in looking at costs in a variety of different kind of processes, actually dating back to a career that began outside of biotechnology. So maybe I'll hand that off to Britton to talk a little bit about that manufacturing facility and how we see that contributing to our ability to deliver our product to as many patients as possible.
Yeah, thanks, Andy. Umoja's philosophy of getting this drug product out to as many patients as possible really grounded the concept of owning our own manufacturing capabilities. And so we built a facility outside of Boulder, Colorado, and we're able to scale. In our view, we'll be able to scale to a point where we can produce thousands of vials per batch. With costs that are similar to MABs or monoclonal antibodies, under $10,000 a dose, and potentially in that $1,000 to $3,000 range at scale. And you compare that to the COGS on autologous CAR Ts, which are on market today, over $250,000. So it's a significant step change, I'd say, in where we can go in terms of patient access and scalability. And we're really focused at the manufacturing facility, just like any large scale manufacturing plant, whether it's in biologics. And as Andy mentioned, I have kind of an oil and gas background, worked at oil refineries, but it's the same concept of how do we optimize and keep costs down and be able to get our cost per vial to a place where our COGS are low and we can produce a significant number of vials to get this out to patients and make a significant impact on cancer and potentially in autoimmune indications.
And actually, from the very beginning, one of the other things we did is we had folks in the manufacturing facility who understand scale talking to our basic scientists who know the vector technology. Academic scientists, and I'm one of them, we have a tendency, you want every bell, every whistle. It's always more exciting to do, really, really cutting edge things. But if you can't deliver it to every patient, or especially, or at a reasonable cost, you really haven't kind of met the goal of the company. So we had people talking from the very beginning, let's build products that are set up in a way that they're modular and something that our colleagues in our manufacturing facility could build. That started literally from day one when Umoja was founded. And I think that was based on the insight. I've been involved in founding companies before. Our other scientific founders had founded companies before, and they saw the failure points that emerged over time by not taking that approach. And that's one of the reasons we incorporated that very early into what we're doing at Umoja.
And I think also, when we think about, pharma, biotech kind of business model, right? Where the kind of bulk of the cost is in this kind of R&D process, which is actually really linear, comes up with like a kind of unipotent end kind of product. and then manufacturing, it was the kind of in the end. But here, because your product is kind of really, it's a vector itself, it's a kind of platform which enables you to go after many different things with small kind of modifications. So if you kind of invest primarily in just developing that kind of platform tech side, it's really about manufacturing to give you the scale, because this often kind of having the kind of modular product gives you the scale which the kind of old small molecule, kind of even the biologic like drugs could not have, right? So I almost see this as a kind of paradigm shift, how we should be thinking about, pharma kind of business model, and where the things are going.
Yeah, exactly. So a great example is we have our two lead products. Those are built on the VivoVec platform technology. This way that we've developed to deliver genes to tumor fighting T cells in vivo. And what we can do is they target specific proteins on the surface of one type of cancer. We have whole teams working on putting in a different recognition moiety. So it's almost exact like the particles that we will generate for each new product on the outside will look the same, because they'll be those particles are engineered for delivery to T cells, but the programming that's delivered to that T cell can be completely different, and will allow that instead of directing that T cell at one kind of tumor, it will direct it to another kind of tumor. And to your point, as we start to look at different types of tumors, more sophisticated programming may also be something that we start to deliver. So the modularity of that delivery platform is absolutely kind of I think of that as a second level of scale. So the ability to manufacture inexpensively the platform particle, the delivery particle, that's one part of it. But the fact that it's modular and we can take all of that effort and all of the manufacturing expertise that we develop for one product and apply it to basically building the next product and the product after that, that's a second level of scale that, to your point, is is kind of a new way of thinking about drug products.
Yeah, I mean, even going back to your analogy with iPhone, it's like almost, the vector is the iPhone, and you're just adding the new apps.
That was part of my the original thesis for Moja when I was building it and describing it to some of our earliest investors was exactly that. The programming you deliver is the new app, and we'll be switching that out for every single tumor.
And I think also there's important regulatory ramifications here. My understanding that, FDA did came with a kind of new kind of set of rules about the kind of platform kind of products, where if there are kind of smaller changes to already approved product, the trials could be kind of shortened or changed, that they are not taking that long. Because obviously one of the huge things in drug discovery, the trials, the clinical piece is the longest and it takes the most kind of money, and it's the most inefficient part, right? And so this gives you even the way to kind of fight the kind of some of these regulatory hurdles in the time frames and the kind of cost.
Yeah, absolutely. The FDA has come out with something called a platform designation that you can apply for. It has to be for something a little bit like what we're doing, where one part of a drug product is fixed, and you show over several drug products that part of the drug product functions consistently, even though you make changes in other parts. And for us, the outside of the particle is the part that really defines our delivery approach. That part doesn't change between each drug product. It has certain properties and ways that it behaves in the body. And we can show the FDA, and actually are already planning to do this. Hey, when we build products on this platform, they have certain predictable properties. We can infuse them. We know they're going to be safe during the infusion period. It's the programming that's different that we'll go in and be testing that, individually for each product. And that saves you all kinds of money, because instead of for each product having to go through a complete characterization of the way that your particle performs, you can show a demonstration that, hey, the programming is functioning as predicted, and that allows you to avoid having to do some of the toxicology and other types of testing for the particle part of it, for the outer surface part of it. You just have to show that the programming is working as designed. So that's another aspect of the efficiency of developing a platform technology for building drugs versus sort of one by one, molecule by molecule building drugs, which has been more of the paradigm in the past for things like small molecules and and monoclonal antibodies.
Just a thought, since we touched on it. It's sort of a provocative topic, maybe, well, always, and maybe especially now as efficiencies are coming into the health service generally with the new administration. How do you think about, when you are able to bring about such cost declines, how do you think about the go-to-market around those, and to what extent that is conferred ultimately to the end payer versus retaining margin in order to recoup, to do more R&D, to do even better therapies for patients in the future? How do you think about that balancing act? And also geographically, right? Because drug pricing in different geographies is so different.
For me, the most important thing is providing benefit to patient. So if we generate drugs and they have impactful, and they're impactful, right? That patients receive them because of our drugs, they're able to avoid chemotherapy. They have long periods of durable remissions, maybe even significant fractions achieve cures. I think that is recognized as deriving value. And for the most part, you're able to price those appropriately and generate very strong margins and returns that allow you to continue to work on your product. But you've got to deliver that transformative impact. That's what allows you to continue to do that. Where I see a lot of pushback against pharmaceutical industries is when the drugs that are developed don't achieve, they achieve incremental impacts, yet the cost that then is asked for, the price that's asked for is very high. So we're, at least what we aspire to at Umoja is that transformative model. We're for those, and again, it comes back to only 1 in 5 patients is getting these drug products. If we can allow CAR T-cell mechanism of action to be delivered to that other 80%, and we really transform their lives by allowing them to avoid some of the side effects of chemotherapy and things like that, we think we can build a fantastic business, deliver impact to patients, have reasonable pricing, and still be able to achieve the margins that allow us to continue to innovate. That is the American way, this idea that, and remember, all of those innovations will be patent protected, but eventually they'll come off patents, in which case they'll be available to every other pharmaceutical company who will now start to generate their own drug products and put even more price pressure on it. So I think this if you look at innovation across medicine, the vast majority of it has come from really the American model, where you have the ability with a transformative, innovative drug to charge a lot, and that allows patients now to benefit. But one of the things people never think about is that the drugs we're taking now were transformative drugs 40 years ago, and now they're incredibly inexpensive. And so the drugs that we're developing in 30 years will be inexpensive, and the next generation of really innovative drugs will be, what we can only imagine, I think, incredibly exciting and sophisticated ways that hopefully will allow people to extend not just, not just surviving cancer, but maybe even extending their health spans and achieving other types of effects on patients' lives.
That's a great segue. I mean, I think we're very aligned in terms of, with those kind of levels of cost declines, there's plenty space to do the right thing by the patient first and foremost, also do the right thing by the whole system, and then make margin and just carry the baton forward and the science forward. And as you say, there's this sort of rolling thing with the biosimilars and the generics, and steadily making progress. The other thing that often gets missed from some of the debate on drug pricing is, as you say, when you have really effective therapies and even cures in some cases, or things that work essentially like cures, and we've been looking at this in the context of gene editing, the amount of saving of other costs downstream is just enormous, because pharmaceuticals is only something between 12 and 15% roughly of all healthcare spending, whereas of course all of the other stuff, not to mention that patients have a terrible experience if they're coming back to the hospital for years, and inpatients and all the rest. But that's a huge amount of expenditure, all the estate and insurance and all the other things that's just not directly hitting the bottom line for the patient's health. So then maybe that's a good segue into, how big could this be in terms of, you mentioned for a second there, I think autoimmune diseases and other places, obviously, with immunosenescence and the degradation through time, unfortunately, in everyone's life, of the immune system. It's such a huge part, a central part of of aging, and the flip side being, could be a central part of longevity or treatments for age-related diseases in cancer, also infectious diseases, but also, there's pretty good evidence, even neurodegeneration, certainly autoimmunity, and probably more broadly, as we learn more and more about the interplay between the immune system and the rest of the body, it becomes quite clear that along with metabolism, it's one of the central axes of aging or of degeneration. How do you think about that? How big could this be? Where else could it be applied? Is there a clear stepwise path to learning more about how this kind of approach could be used more broadly? Or is it very clear that, really the central thrust will be in oncology for the time being?
Certainly the near-term thrust is oncology, but to your point, there are some really exciting applications in autoimmunity as well. Your audience may not be aware. So over the last, I'd say 5 years, one of the most exciting approaches to treating autoimmunity that's come out has been the concept of an immune reset. And it kind of makes sense, which is if we look at patients with the kinds of the autoimmune diseases, usually they're perfectly normal for some period of time, and then something happens and that causes an immunologic attack on healthy tissue. And actually, it is an incredibly broad range of disease. Certain types of diabetes are clearly autoimmune diseases. The obvious things like rheumatoid arthritis is an autoimmune disease. There's a disease called lupus where you have attack on kidney and, kind of broad attack on organ systems and vessels. So these are very frequent parts and more frequent as we age. And it may be that immune senescence is a little part of that. That is, if you're my age, you've already started to experience, hey, not everything's working the way it used to when we're in our 20s or 30s. And I think the immune system's no different. It becomes, not as specifically targeted. The ability to have a strong immune response so you can recover from a viral infection, but then terminate that immune response and get back to your normal state of feeling good. Those are just not the same as where things were 20 years ago. For autoimmune disease patients, what clearly happens is you have a durable autoimmune attack on a healthy tissue. And what somebody observed was, hey, if you go in and you wipe out a certain class of immune cell, which is the one kind of immune cell that we know undergoes a genetic rearrangement as part of the process to help you fight off infections. But if you wipe out this class of cells, you're actually able to reset the immune system and create drug-free remissions where you have patients with severe autoimmune disease that normally require multiple drugs to control these immune responses are now off drugs for in some cases indefinite periods of time. They're still off drugs 2-3 years after these immune resets. So those are certainly, and this kind of immune reset has been achieved with CAR T-cell therapy directed against this type of immune cell that undergoes this genetic rearrangement. And absolutely, those are things that we aspire to begin to try and treat as well. The reason for starting in oncology is the established role of CAR T cells in oncology when the company was founded, and the fact that the risk-benefit ratio, it's a new modality, and so regulators and all of us should be careful about how we apply a new modality, ensure that it's safe and beneficial before we begin to move on to diseases that don't have that stark, hey, cancer is going to, end this patient's life in the very near term, don't have that stark risk-benefit ratio. So autoimmune disease generally plays out over a bit longer period of time. So we are going to move into it. It'll follow what we do in oncology, but we do think there is an opportunity for really delivering some transformative benefit in that area as well. Longer term, Charles, that's a conversation to have over maybe a glass of beer or some bourbon. But one can certainly imagine that there would be opportunities in patients who might otherwise be healthy to deliver genes that would restore a state of immune reset and potentially avoid some of the aging-related, more kind of vaguer symptoms that we all encounter as we age. In the form of an immune reset, in the form of eliminating senescent immune cells that create, the inflammaging phenotype that, sadly we experience every morning probably now. So.
Sounds good. I'll make an alcohol-free beer if we're going to discuss longevity.
Yeah.
Otherwise, I'll.
Sounds good. Yeah.
All right. Thank you.
I actually, I just wanted to kind of close the circle here. I think it was great for us to kind of really go think about the future. ARC is all about the kind of future and where it will take us. But maybe, let's also kind of close it with the kind of near-term future for Umoja and just talk a little about the kind of current pipeline. And so, we know that what exists already in the market is the CAR Ts for the CD19, but obviously a lot of different issues there. And so what are some of the kind of pipelines highlights you want to share with our audience and kind of some of the possible catalysts in the near future?
Yeah, so near term for us is we have our 2 lead programs enrolling. So it's beginning to treat patients in the clinic. Our initial products are for blood-derived cancers, and we're looking to extend that into a form of cancer called myeloma, I would say initially. And then also, and you alluded to it at one point too, into I'd say a general way of treating solid tumors. So solid tumors are tumors that derive from other types of tissues than blood. So that could be liver cancers or colon cancer or breast cancer or lung cancer. They have unique properties versus the kinds of cancers that are blood derived and require again, to the point you were making, some more sophisticated type of programming. So we're working on developing that programming and ways of targeting those solid tumors. Those are things that we would see coming out probably in the 2026 timeframe. But near term, it's blood cancers, myeloma, which are areas where we have existing CAR T cell products, so we know that the CAR T mechanism of action can be transformative, and we're bringing the goal is to bring our products out, expand access, build the manufacturing base, and then move into solid tumors. I'd say that's the sequence for how I see Umoja developing over the next couple of years. And we're obviously excited to have ARC Invest on board as one of our earlier shareholders and investors, and look forward to having you along our journey as we continue to do things that we hope will change the practice of medicine. That's really I retired from my academic position 5 years ago to go all in on this, and I couldn't be more excited about the future for us.
Well, great. So thank you both for being our guests, and we are looking together with helping you guys build this amazing future ahead of us.
Likewise.
Thank you so much, and we're very excited to see what these technologies bring for patients. So please let us know how we can help, and look forward to the journey.
Thank you for listening to this episode of FYI, the For Your Innovation podcast. If you enjoyed this episode, please hit that like button and make sure to leave a comment down below letting us know what you thought. And if you haven't already, please make sure to subscribe to the show and follow us at ARC Invest on your favorite social platforms so you stay up to date whenever we drop a new episode. ARC believes that the information presented is accurate and was obtained from sources that ARC believes to be reliable. However, ARC does not guarantee the accuracy or completeness of any information, and such information may be subject to change without notice from ARC. Historical results are not indications of future results. Certain of the statements contained in this podcast may be statements of future expectations and other forward-looking statements that are based on ARC's current views and assumptions and involve known and unknown risks and uncertainties that could cause actual results, performance, or events to differ materially from those expressed or implied in such statements.